the auditory information gets processed in a very basic way in the auditory cortex, but additional processing happens outside of the auditory cortex. So you can see here the auditory cortex is here in the temporal lobe. So, and of course there's one here on the right side and one on the left. But these colored areas are also involved in processing of sound. And they are divided up into areas of the brain that process what we are hearing. And these are shown here in green. And notice that these are primarily in the temporal cortex and a little bit further back here in the towards the occipital lobe. So they're more on the lower parts of the brain. In contrast, the areas shown here in red are involved in processing the location of sound. And so we basically have like two clusters, a cluster of areas involved in figuring out what we are hearing, and a cluster of areas involved in figuring out where those sounds are coming from. So we have what and where areas in the brain. And we'll see that the brain does the same thing for vision when we get to the chapter on vision. So the what pathway is also referred to as the ventral stream. Sometimes it takes a ventral route from the auditory cortex down to lower levels or inferior levels of the temporal lobes. In addition, part of that what processing includes something called the secondary auditory cortical area. So the information from the cochlea first arrives in the primary auditory cortex, where it's analyzed according to pitch, and then it gets sent to the secondary auditory cortex, and that's where more complex processing of sound, like for example speech and music, happens. So the primary auditory cortex does not do speech processing or even music processing. This type of processing happens afterwards. The where pathway is also sometimes referred to as the dorsal stream because it goes towards the dorsal side of the brain, the top, and it goes primarily to the parietal lobes and then forward to the frontal lobes. And so the what and where areas are kind of distinct. One more ventral for processing what the item is, and the other one more dorsal for processing where the item is. Now, in terms of hearing, we're going to see that we need to figure out how the brain categorizes and interprets pitch. How does it figure out the pitch of a sound? There are different possibilities. The first one that was developed is called frequency theory. And frequency theory is really beautifully simple. Basically, whatever the frequency of the sound is the number of times per second that the auditory nerves are going to be firing. This is also known as telephone theory because it just relays, like a telephone, the signal being created by the sound itself, the frequency of the sound itself. And so early researchers did find that the auditory nerve responded in sync with the frequencies being heard. So this works great. There's a problem though. The auditory nerve, like any other nerve in the body, is not able to fire super fast. There's an upper limit. That upper limit is about 500 times per second. So I'm going to go ahead real quick. And it's because neurons can't fire, say, 1,000 times per second. So they can't fire fast enough to allow frequency theory to work. And the reason for that, remember, is the absolute and the relative refractory period. But even the absolute refractory period inserts a couple of seconds here between action potentials. Volley theory was developed, and this helped to deal with the fact that it's impossible for a single neuron to fire more than 500 times a second because of the refractory period. So the way that volley theory works is, instead of a single neuron firing, say, 1,000 or 2,000 times a second, a group of neurons take turns. And each of these 5 neurons in this example is firing, once every 5 times. Together, they represent each peak in the frequency of the sound, and they're firing once. So cumulatively, they are firing one time for every sound wave that's passing through per second. But they're not doing it independently, they're doing it collectively, and so this is called volley theory. Volleying fails with sounds beyond about 5200 hertz, so this also has an upper limit to the number of neurons that can work together and how fast they can fire. And so this gets us to place theory. Place theory has to do with the location or place of where the hair cell is in the cochlea. And it turns out that the hair cells on the cochlea respond to only certain frequencies of sound, and that they are very organized and go from the highest frequencies here. So the stirrup or the stapes would be right here in this area. And so this area of the cochlea has hair cells that respond to the highest frequencies. And as you move down into the cochlea, you can see that the frequencies that the cochlea responds to go down until you get to the very end, and that's where the lowest frequencies are responded to. So that's place theory, that the place along the cochlea is able to tell other parts of the brain what the frequency was, because if it was a hair cell from this zone, we know then that the sound would be 10,000 hertz. If the neural signal came from this area of the cochlea, then we would know it was a 3,000 hertz tone, and this one would be perceived then as a lower pitch than the 10,000 hertz. Here's a demonstration of how the cochlea works according to place theory. So we're going through the auditory canal, and there's the cochlea, and it's going to unwind. And you can see that the different frequencies are creating activity along different areas of the cochlea. And so this tonotopic map is due to the varying flexibility of the basilar membrane where the hair cells are. And when we say tonotopic map, it's really a map of the different frequencies or tones laid out in order. And basically what we see is that if we can think of how the basilar membrane is laid out, so there we have the cochlea, the membrane there, the little hair cells. This area here where the stapes are, the stirrup is, this area is the base, and the far end of the cochlea is the apex. And so the base has the hair cells that respond to the high frequencies, and the apex has the hair cells that respond to the lower frequencies. So it goes from high to low. And we know that the cells in the auditory cortex are also arranged in the same way. So they are also tonotopically organized. And in fact, the cells are tightly tuned to specific frequencies. And so if you record the activity of a given neuron in the auditory cortex, you'll see that it'll basically have a preference for a particular frequency of sound, and it'll respond to that sound even when it's at very low volume. And so we're seeing here that a given hair cell has a preferred reaction to, say, 700 hertz here, and I'm not sure what this is, 11, 12, 1300 hertz here, and so on. And so each of these different neurons has a narrow range of frequencies that it responds to, and that allows the processing of frequency. These tuning curves, where there's this really narrow range of frequencies that the hair cells respond to, is due to something called lateral inhibition. Lateral inhibition is kind of what it sounds like. There's inhibition of the neighboring neurons. So if this particular neuron right here that really likes to respond to 1300 hertz is responding to 1300 hertz, it will also then inhibit the neurons next to it. So if it's here in the auditory cortex and it's picking up that 1300 hertz sound, it will inhibit the neurons to the left and to the right, making sure that there is a precise neural signal for just that one frequency. And so because we can't use frequency theory to explain how all sounds are processed, we have frequency and place theory. It combines the two theories for us. Frequency theory governs how we respond to frequencies under 200 hertz, and place theory does everything else essentially. So for 200 hertz and lower frequencies, individual hair cells fire at the same frequency. So if it's 150 hertz, they'll fire 150 times per second. The number of hair cells that are doing that probably conveys the sound's loudness. But for louder sounds and for higher frequency sounds, place theory is used. So for everything greater than 200 hertz, place theory is what determines the pitch that we hear. We also have learned some really interesting things about how the brain processes complex sounds. So there's something called the cocktail party effect. It allows us to follow an auditory object even within a complex sound background. What that really means is you're at a party, there's a bunch of people talking, music on, all kinds of noises, but you're able to follow one conversation. And that one voice, for example, would be the auditory object. The way that works is that higher level neurons actually go back to the basilar membrane, down into the cochlea, and they influence the outer hair cells and make them ignore frequencies from voices and other sounds that you're not trying to pay attention to. So this is called feed backwards. And this is kind of interesting because it's not something that is seen that often or that easily in the nervous system, where higher level neurons from the cortex go down to areas that have been processing that information earlier, and they influence that processing, that early processing.